What is the clinical trial design to evaluate plasmalogen augmentation for the prevention and treatment of neurodegenerative diseases? So we've done an extensive amount of research on plasmalogen levels epidemiologically in aging, how they decrease as we age. We know the distribution in the population, how many people have high levels versus low levels. We know that people with high levels have a dramatically reduced rate of dementia in the population. So we have a large amount of knowledge about this. We also have the distribution levels of plasmalogens in the brain that we know. What we don't know is what is the optimal level, okay? Because the highest levels in the natural population still might not be optimal. We may want to be even higher than that. So part of the clinical trial process is to understand the augmentation process. We know in animals that getting 2 to 5-fold increases of plasmalogen levels in the blood is easily achievable. We know that approximately 2-fold increases in the blood are very neuroprotective in animals. But now we have to study the human animal. We need to understand, so the clinical trial design is, there's a baseline assessment to understand what plasmalogen levels are at the beginning. What is the clinical characteristics of the individuals before they enter the trial. Then the first step will be providing an increasing dose of plasmalogens. We want to understand how much plasmalogen precursor is needed to reach a target dose level, and we will measure and find out whether or not there's any kind of side effects. Side effects haven't been seen in any of the animal studies. We know the DHA and other types of molecules like this have been studied very extensively with no side effects at all. So we're not anticipating any of that, but we will measure that and keep track of that. The purpose then will be to find what is our optimal titration of plasmalogen levels in the blood. Because the purpose here is to prevent neurodegeneration and to maintain optimal neuronal functioning for the longest period of time. So that's the first part. So each person becomes their own control in that model. And then it's a matter of time. We start with an elderly population that we know in advance what degrees of decline should occur, and we compare that with individuals on the therapeutic program. We'll also look at improvements. People that already have decreased functionality, how much and when, or if we get improvements in the neurological function. So the clinical trial design is a community-based model. Our goal is to benefit the individual users. So it's not just about the plasmalogen augmentation. It's other aspects of their health will also be optimized, whether it's their diabetic health, whether it's their cardiovascular health, their high blood pressure, their other drug utility. So the whole purpose of a community-based trial that is very open-ended is an adaptive clinical trial design so that we have the ability to measure cause and effect in a large enough population to have a high degree of confidence of what the plasmalogen augmentation is doing or what it's not doing. And it's being a very open and transparent process that everyone can see, because the purpose of this is really to engage the community in a program that is going to be relatively inexpensive for users to adapt the product into their lifestyle when the trial is done.